Hello. One listen to me.
Okay. Hello, test.
Hello. So we need.
Okay.
Good morning.
Well, good morning. Good morning, Professor.
Yeah, good morning.
Okay. Good morning, ladies and gentlemen, audience and seminar participants. Welcome to this meeting. We proudly present the seminar and discussion on the topic tips and tricks how to write a scientific article for High Impact Factor Journal 2020. It's been a great honor for me, Ida Ayu Wimala, to be here and welcome each and every one of you in this meeting. Ladies and gentlemen, over the next few days, this meeting will provide you plenty of opportunities to discuss, share knowledge and insight. Before we process the program today, we would like to express our sincere gratitude to Excellency Head of Aquaculture Master Study Program, Professor Dr. Insinyur Mahfud MSc. Excellency, a speaker from National Pingtung University of Science and Technology, So Young Hu. Welcome, Professor. And the Honorable all lecturers, the audience, and seminar participants. Okay, the next agenda is a group photo session. To all seminar participants, please turn on the camera. Please turn on the camera. Okay, let's get ready. The photo session will start on the count of 3. 1, 2, 3. Okay, once again. 1, 2, 3. Okay, thank you for your attention. Excellencies, our distinguished audience and seminar participants, without further ado, we would like to invite to deliver his welcoming speech. As head of Aquaculture Master's Degree Program, Professor Dr. Insinyur Mahfud MSc. Professor Mahfud, time is yours.
Thank you, MC. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, everybody. The Honorable of Professor So Young Hu from the Department of Biological Science and Technology, National Pingtung University Science and Technology, Taiwan. And also all of the participants and my student from Aquaculture Master Degree Program. Firstly, allow me please to thank to Professor So Young Hu for sharing and guest lecturing today on November 20. And also secondly, I would like to express that writing and publishing research result is the obligation of a student to complete the study in Brawijaya University. Secondly, write and publish research result to be more usable for the people, and also for copyright recognition, and also for property rights, and I think many other functions. We know that this activity is not easy to do. It's my mention is writing and publishing the result of the research. So we invite Professor Saoyang, who belong to his expertise, to give lesson learned about tips and trick how to write a scientific article for a high impact factor journal. So finally, I hope that the student can attend this guest lecture well, pay attention, and can take the best lesson from Professor Saoyang Hu from this guest lecture today. Thank you very much. I turn back to MC. Dayu? Dayu, you still mute. Unmute first.
Oh, I'm so sorry. Thank you, Professor Maftub, for the welcoming speech. The next agenda is Excellencies, ladies and gentlemen, and audience. Now we come to our main program, who will led by our moderator, Talita Amanda. Talita, time is yours.
Thank you, MC. Good morning, ladies and gentlemen. May I have your attention, please? The lecture is about to begin. Is my voice clear enough? Is my voice clear enough?
Yeah, enough.
Yes.
Okay, very clear.
Okay, thank you. First of all, I would like to welcome you all to this lecture. My name is Talita as moderator for today. Before we begin, please let me read the CV of Professor Saoyang Hu.
Share screen Mbak Dalan. Mbak Dalani, please share screen Professor Yang Hu's CV.
Today we are pleased to welcome Professor Saoyang Hu from National Pingtung University of Science and Technology, Taiwan. Professor Hu finished his education at Institute of Microbiology and Biochemistry, National Taiwan University. Professor Saoyang Hu's specialty is applied microbiology. I'm sorry, maybe you can share screen. Okay, I will continue. And this is Professor Saoyang Hu's scientific publication list within 5 years. Today, Professor Saoyang Hu will present about tips and tricks how to write scientific article for a high impact factor journal. Without further ado, I would like to welcome Professor Hu to deliver the presentation. Please welcome Professor Saoyang Hu. Time is yours.
Okay. So, can you listen to me? Is the
Yes,
voice
very
clear?
Yes, very clear, sir.
Yes, sir.
Okay. Okay, so right now I turn off the video and share my screen to you later.
Okay, sir.
Okay. I make sure that all of you can see the screen. Hello?
Hello, Professor. It's already
Yes.
appeared.
Yeah, I know, but I mean, can you see my screen?
Yes,
Yes,
Professor.
sir.
We can see the screen.
Okay. Okay, first I would like to thank Professor Matu giving me the opportunity to share the talk. Previous, we already have one lecture in November 2, and today the lecture title is Immunological Molecular Pathway of Bacterial or Virus Infection on the Zebrafish Model. So I think that when we talk to the zebrafish, we should know what is zebrafish and what advantage the zebrafish have. So today, our night, we will briefly introduce the zebrafish and the advantage of the zebrafish animal model. And we also talk to the inner immunity systems in fish. And after we introduce the inner immune system, we will introduce some case. For here, I will introduce the hepcidin is an antimicrobial peptide, and we use hepcidin expression app because when we produce the hepcidin, we undergo some problem. So we try to solve this problem, and we use another system to express the antimicrobial peptides. And finally, topic is scientific writing for impact publication. Actually, to talk this topic, I feel uneasy because I also want someone to teach me how to publish the high impact journals.
Yeah, sure.
So anyway, I just think to share my experience to you.
Yes,
okay.
Thank you.
So we back to our content. The first one, I would like to introduce zebrafish, Daniel Reynolds. Maybe because I think all of you know fish like me. So when I introduce the zebrafish, maybe all of you already know that. Okay, the zebrafish Daniel Reynolds. When you see the photo, you can see that after the fertilized egg making 30 minutes after the fertilization, one cell stage embryo he can start to development, and so you can see the cleavage and shoring and shared and antibodies just 2 days of fertilization, the zebrafish can hatching out. So in the right side, you can see the embryo is transparent, okay, and quickly development. That's why the zebrafish is common used in developmental biology studies. Okay, and after the 3 months culture, the zebrafish can maturation. It means it can making and get the offspring, can get the fertilized egg. Okay. So when we use zebrafish to study no matter the human disease or developmental biology or the aquaculture applications, have some advantage. For example, the culture zebrafish quite small, so no need large space to culture the fish. You can see the photos below. It's my fish room. Just a small space you can capture the zebrafish, and compared to the mouse system, the cost is low. Okay, and you just see the photo, the life cycle, just 2 or 3 months, it can maturation, and it means one generation cycle, just 2 or 3 months, and after the mating, okay, one pair of zebrafish, one time springing, it can get 200 to 300 eggs. Okay. And in zebrafish, actually, some skill already well established in zebrafish. For do the transgenic fish, we use the microinjection or electroporation. Actually, the skill already maturation. And transparent embryo, it's easy for observation. Some like the GLP fluorescence proteins. I think it can be used as a biomarkers to trace your target protein, whether it is expressed or not. In fish, we also use some gene lockdown technique like the morpholino or siRNA. I think later I will introduce the siRNA because later when we introduce the case prevent we use some siRNA skills. Okay, and in the fish also can easily use the chemical mutagenesis. It means many mutants you can get from the fish. So you can see the photo below. In here, if you get some mutants and the mutant is similar to human disease, so you can use the debug fish to high throughput screen the drug. Maybe the drug can use the therapy the human disease. So in debug fish, one part research they use to high throughput screen drug. Okay, and the most important when we use debug fish to study the human disease or the developmental studies. It's based on the debug fish genome because debug in 2013, I think the genome sequence already finished. So it means any genes you want to analyze, you want to start, you can get the gene information from the database. Okay, so because we will use some transgenic fish to study the immunological functions. So here's I briefly introduce the transgenic technology in zebra fish. Okay, you can see the slides. That transgenic animals. I think a transgenic animal it means the transgenic protocol is almost similar in all animals. Okay, no matter in fish or in some mammal animals. Okay, the concept I think is the same. When we establish the transgenic animals, I think two technology involved in this study. One is a recombinant DNA, and one is a gene transfer technology. For the recombinant DNA, I think that everyone understands when we want to express a target gene. For example, you clone. If you clone the heterologous genes, maybe it's a hormone or the enzymes that's you want. You clone from other species. Then you will use the resin enzyme and ligate into the vector. Get the expression plasmid. So in the expression plasmid, you can use the promoter to trigger a gene expression. Gene expression. The most important is the promoter. If the promoter is a strong promoter, then your target gene can express strongly. And sometimes the promoter maybe it's a tissue specific promoter. So it means your target gene can express in the certain tissues. If the promoter is a muscle specific promoter, it means your genes can express only in muscles. So after you get a plasmid, expression plasmid, we can gene transfer to the embryo. So in the video, you can see the zebrafish embryo. In my lab, we use two ways to gene transfer. One is a microinjection, and one is electroporation. But for high to increase the high injection rates or gene transfer rates, we use the microinjection. And the way is we put the recombinant DNA into the noodles. The noodle scale is 0.1 or 0.5 micrometer noodles, then inject to the embryo. Okay, after inject, then we culture the embryo. If the embryo grow up and become a fish, so you can get the transgenic fish. Okay, so when we call the transgenic fish, it means the first thing, the expression plasmid integrate to genomic DNA, and second is the transgenic animal or transgenic fish, the expression cassette, it can generate transmission to the next generation. If the transgenic fish can perform the two things, we call it a transgenic animal. So remember, the first thing is expression cassette integrate to the genomic DNA. Second, the expression cassette, it can generate transmission to the next offspring. Okay, and I think 20 years ago, at that time I still a PhD student. At that time, when we do a gene transfer, if we want to establish a transgenic fish, it's quite difficult, because at that time, every day you will find your research work just injection. Because it's difficult to get a transgenic line. So at that time, when we do this kind of experiments, we always one time we inject maybe 1,000 or 2,000, 3,000 embryo, but you get nothing, because all the inject embryo not transgenic, because your expression plasmid not integrate to the genomic DNA. But until 2004, Japan scientists, I forget the names, they established the total transposon systems. So here I introduced the total transposon systems. Because this transposon systems established, it's quite easy to establish the changing lines. Okay, so here's I briefly introduce the total transposon. So it's first isolated from the Japanese metaka. So let's call the total the transposon of original lipid. So it's the first text to name the transposons. So it's fine that in Japanese metaka, elements that can encode transposes, and the transposes can recognize transposon element. Then he can cut transposon element and random integrate to the genomic DNA. So these systems right now it widely used in the butterfish, linapers, or the mouse ES embryos, embryonic stem cells. So it means it's widely used in different species. But in the butterfish, how to establish the transgenic fish? We prepare a messenger RNA. We call a transposase messenger RNA. With the construct recombinant DNA contain a transposon element. So we inject the mRNA and recombinant DNA into embryo. It's a plasmid DNA. The plasmid DNA, the green arrow means your expression cassette, and the red lines are here. Okay, it means transposon element. So when you co-inject transposase messenger RNA and recombinant DNA into the embryos, the messenger RNA can translate as a enzyme transposase. Then the transposase can recognize the transposon element. Then it cut out the expression cassette. Then integrate to the genomic DNA. So we can design the primer in the flaking of expression cassette, out of flaking expression cassette. Then it's quite easier to make sure whether your integration is successful. If successful, we can do the PCR. We can amplify a small band. It's around 250 base pairs. The Gen 9, the successful rates so far when we test the successful rates, if you inject the 100 embryos, almost 50 embryo is successful. So it means right now when you establish a transgenic fish, it's quite easy. Okay, and because establish transgenic fish is quite easy, it's easy to study many topics. Okay, so next we introduce the fish in immune systems. I think in November 2, I already introduced this part, but here I think I brief remain again. I think the fish like the mammalian is the same. We use we call the innate immunity against the microbial agents. And last time I already introduced when the patient invest to the cell or invest to the host, our immune cell like the macrophage or neutrophils, they will engulf the pathogens by the phagocytosis. And during this period, some cytokine or interferon will produce to activate cell, immune cell itself or other immune cells. And during this process, also some component will screen, for example, body or cell activities. But all the phagocytosis this stage is triggered by the receptors because the receptors on the surface of immune cells, it can recognize the pathogen. So receptor act an important role. Can you listen to me?
Yes, Professor.
Okay.
Yeah,
Okay.
very clear.
Okay. So the receptor on the immune, the surface of immune cells, it can recognize the pathogens associated molecular patterns. We call PAMPS. That's direct from the bacteria or virus. When it recognizes it, start to trigger the phagocytosis. So I think this part is quite important as the indicator to measure the healthy status. So we can use the slides. The first thing when we to say the prevention disease, it should be prevent the infection of pathogen infections. Like our skin, the first barrier to prevent the pathogen entry to our body. In fish also the same. But even that, I think the pathogen have many opportunity to entry to the host or invent our body or our cells. So our immune cell will use the receptor to recognize the PA and trigger the cytokine interferon screen and induce the innate immunity component like the antibacterial peptides or the complementary component or the lysine activities, something like that. So you can image that in here the receptor also act important role. For example, I think maybe all of you already know the toll-like receptor. Toll-like receptor, for example, toll-like receptor 4, it can recognize gram-negative pathogens. And some toll-like receptors maybe recognize virus or single-strand DNA, or RNA, or double-strand DNA. So different toll-like receptor recognize different PA and PS. So if we understand the immune systems, then we can do our study case. The first study case, I introduced the rice husk silica because we try to develop the rice husk silica as an immunostimulatant. So the topic here is rice husk silica enhance innate immunity in zebrafish and prove the resistance to pathogen infection. Okay, what is a rice husk silica? Rice husk, we call it arch. It's around 20 to 22% rice. Actually, in agriculture, it's we consider it's an agricultural byproduct, and its boundary is available. And arches are often considered a waste of rice during industries. Even right now, some industries try to develop some product, arch product, but you know the arch amount is quite large. So most of arch still usually done in the open areas and dumped on western lands. And these treatment ways are causing the environmental pollution, like photo you see here. So companies are looking for me and trying to cooperate, develop the ultra rice husk as immunostimulators. So it's interesting because in arches you can find silica. It contains high ratios. For example, when we burn the arch, acids around 17 to 20% acid, and acids contains more than 90% is silica. So some study already use a simple procedure to extract the silica. Here you can see the silica nanoparticles. It can be recovered or purified from the arch by a simple some chemical process. It means that it can easily get the silica. We call the arch as silica. It's a low cost and easy get the materials. Review that we find the silica code. It can when it means when the host they treat the nanoparticle silica, it can induce some innate immunity through the toll-like receptor signal pathways, and cause some information. So it means if the host or if the fish is exposed to the high dosage of the silica, it may have damage or toxicity. So in our studies, if we want to develop silica as immunostimulators in agriculture, we should determine which dosage is safe. Otherwise, if we treat high dosage, it may cause damage to the fish. So you can see here it's we extract the silica nanoparticle from the rice husk. Okay, after that we extract the nanoparticles. We use some SEM to observe the surface. So you can see the primary structure is a hydrated silica. It means it has many pores, and the size is around the The size is less than 100 nanometers. After we get the silicon nanoparticles, we prepare as a solution and treat the debug fish. And here we treat the debug fish embryo. Okay, in our experiment, we divide two parts. One is debug fish embryo. One is debug fish geometry. Okay, in debug fish embryos, when we treatment different dosage, control it means without the RHS and the different dosage. You can see from the table when we exposure to the RHS 3-day or 4 days, you can see that if 200 it causes severe damage. We measure the survival rate, hatching rate, and malformation rate. You can see that all the number is lower compared to the control or other dosage. So it means if the then the 200 microgram per milliliter, it will have toxic to the debug fish embryo. And the same situations in debug fish geometries. We also exposure to the geometry debug fish. The same, you can see more than 200. It causes larger molarities. In 200 concentration, 200 microgram per milliliter concentration, the number compared to the control, no significant difference. It means in this concentration, it's safe. Based on the data, so the rice hard silica less than 200 microgram per milliliter is the optimal dosage for treatment debug fish. And after we treat the debug fish, we find we treat 7 days for 7 days. And after 7 days, we analyze the immune parameter. We analyze some immune parameter genes, like the it's a cytokine genes, like the interleukin-1 beta 6. You can see here the toll-like receptor 4, complement body or lysosome enzyme. You can find in the 20 some indicators in 20 already increase compared to the control. All indicators in 200 gene expression is significant increase compared to the control or the other concentration. So it means in 200 microgram per milliliter is optimal. In this concentration, it already can modulate the immunity. Then we do a challenge test. Okay, so here's we do the two concentration, 20 and 200, and test the two pathogens, Aeromonas hydrophilia and Staphylococcus aureus. Okay, you can find if we exposure to the 200 microgram per milliliter RHS, it can enhance the disease resistance to pathogen infection. You can see the survival rate compared to the 20 or the controls can increase. So based on the data, you can see the 20 at 20 this concentration is no use. It can increase the disease resistance. So in this part project now because this project we cooperate with the bio companies, so they already apply the patent and develop the rice hard silica as a modulators in aquaculture, just one bottle solution. But I think this product not directly used in large field aquaculture. All of their product are used in lava fish or in ornamental fish. Okay. summary, the silica particle with a multiple pro structure was required from the rice husk by simple process, and the debar fish is put to the rice husk silica. Those lower than the 200, no damage to the debar embryo and the jumery debar fish. And the safe IHS dose enhanced immunity against infection without toxic effect. And this result suggests IHS may be developed as an immunostimulator for the improving health states in agriculture. Okay, this is the first part I introduced. And the second part, I introduced the antimicrobial peptides. And here I will introduce the hepcidin. This antimicrobial peptide. We see what is the antimicrobial peptide. I think all of you maybe already know. Here I briefly introduce. MP is a major component of inner immune defense of fish against pathogens. And several papers already mentioned MP only the antibacterial activity. They also can have the immunomodulation functions, so which enhance the fish can increase the host defense against the pathogen infections. Okay, but when you check the antimicrobial activity from different species, you will find their functions seems different because their amino acid or the structure different. So their function, antibacterial function, is quite different. So based on their specific structural characters, the antibacterial, antimicropeptide, they can divide a different class. For example, here I show 4 class. The first one's linear-based peptides with amphipathetic alpha helix. For example, scoping A, BD, that's from the insect. Pyricidin from winter founders is with the disulfide. Dosomycin or defensin, that's from the fruit flies. And some antimicrobial peptides, MP, they reach the purine or glycine. For example, here's doxocin, also from the fly, or belecin from the cattle. And the last one is from the plant. Okay, the glycine-rich antimicrobial peptide. Why the antimicrobial peptide can kill bacteria? The defense mechanism of MPs, I think, is first it attached to the cell membrane of pathogen. They grow the peptide. They form a pore within the membrane. So it causes the cytoplasma flow out, so the pathogens dies. So here I show the video. So the videos show the mechanisms of antimicrobial peptides. So when we see the actions, I think the MP it major act on the cell membrane. So sometimes when we treat the MP bacterial cell with the MP and see the surface under the electron microscopy, we can see the cell membranes was disrupted. And today the topic we will introduce the hepcidin. Hepcidin is a small cysteine-rich condition antimicrobial peptide widely exists in diverse species. Okay, in the beginning, the hepcidin is named as a liver-expressed antimicrobial peptide because it was first isolated from the human livers. And based on the structures, you can see it with 4 disulfide bonds. So it means that its structure is quite complicated. So when we review the papers, hepcidin cDNA so far it has been isolated from several fish species for the tilapia, salmon, or the chong. I remember several fish species already hepcidin already become but when we even the hepcidin already isolated from the diverse fish species, and we can see that when we analyze or study the hepcidin's functions, we always use the sensitive the hepcidin. That's the problem. Later I will introduce. And when after the hepcidin isolated from the different fish species, and we check the antimicrobial spectrum, you will find that the spectrum is quite different. So it means when you isolate a new hepcidin peptide, you still need to analyze its function. To this point, in our studies, we isolate from a new hepcidin gene from the ornamental fish. Later I introduce. So we need to analyze its functions. Okay, this is our target. Convex chelate is an ornamental fish. Convex chelate is a major ornamental fish species in culture in Taiwan or in South Asia countries. Maybe 10 years ago, since the first transgenic ornamental convex chelate was successfully established in Taiwan, it has become more popular and more attention devoted to the disease resistance. Many people like to capture this kind of ornamental fish, but for farmers, when they capture this kind of ornamental fish, they still face some disease problem. For example, previous in November 2, I introduced several pathogens, freshwater, like Staphylococcus, Aeromonas, these fresh pathogens also happens in ornamental fish. So we isolate hepcidin gene from convex chelate. Okay, we isolate the hepcidin genes, and the gene structure contains 3 exons and 2 introns, and we alignment with different fish species the same. Gene structure, the protein structure, they contain the single peptide and the correct side. And here is the mature peptide. Hepcidin is indicated. It means mature peptide here. The hepcidin we named as ANHEPC. ANHEPC. Detect it's a tissue distribution. Okay, so in the convex chelate, you can see the major hepcidin is expressed in the liver. There's paper published. They also they find the tissues. Kidney also have. Okay, and the intestine, heart, stomach or spleen, this kind of tissues have the hepcidin expression. And after that, we in COVID chelate, we challenge with the LPS, lipopolysaccharide. It's a component of gram-negative strands. After we injections, you can see after we injection in different time points, 6 hours, 12 hours, 24 and 48 hours, you can find the the black bar is a control, and the gray bar is a LPS challenge. You can see that when LPS challenge in different tissue, the hepcidin genes was induced very significant. That means the hepcidin roles in this kind of fish. So we test two parts. First, we analyze its antibacterial activity. Use sensitive hepcidin. I will introduce why we use sensitive hepcidin because it's difficult to get negative hepcidin because it's complicated structures. So when you express or if you want to produce the hepcidin from the E. coli, it's quite difficult. So in the beginning, we use the sensitive hepcidin. We test different pathogen species. Some species maybe relate to the aquaculture, and some relate to the food or the clinicals. So you can see the MIC and MBCs. It can show the antibacterial activity to different pathogen species. And we also see the, We also see the morphology of pathogens after they did the sensitive hepcidin. Okay, you can see this one is is E. coli, and in the left side it's a control. It means without the hepcidin. In the right side, the arrow after treat for remember 1 hour. 1 hour. Okay, you can see here. You can see the membranes is disrupt. All some membrane is atrophied or changed. Okay, so it means this kinds of antimicrobial have the antibacterial functions. Okay, so same we use a sensitive hepcidin test. We also use two pathogens, Staphylococcus aureus and Beneficus. Okay, you can see that when we use this dosage, per fish is 0.1 microgram. Treat the fish first. Okay, then inject. You can see here. If we treat the hepcidin first, you can see the survival rate is a significant increase compared to the control. So it means this antimicrobial peptide, hepcidin, it can use in aquaculture to resistant disease. Okay. So here's I also do a summary. The hepcidin gene was cloned from liver, from the COVID challenge, and the expression during immune response or characterized. The sensitive hepcidin peptide show a wide spectrum of antimicrobial activity in vitro and through the gram-negative or positive pathogens. The important role for the hepcidin in the innate immunity of COVID challenge and suggests its potential application in agriculture for increased resistance to disease. I just say that isolate the genes. We want to study the gene functions, but the gene or the peptide is difficult to produce. Previously, we tried to express the peptide in coli, use E. coli system. But most of the proteins are expressed as an inclusion body. It means insoluble forms. And you know that if you want to express an antibacterial peptide, but the E. coli is also a bacteria. So if it induces the recurrent protein express, the host, the E. coli, will be killed. So it's quite difficult to produce a recombinant, the recombinant hepcidin proteins. And why we need use why we need to produce the hepcidin peptide? Because sensitive peptides, the first one, it cannot completely reveal the structure or biological activity of negative hepcidin. And second, it's a sensitive peptide. The cost quite high. It's unsuitable for practical use in agriculture. And in E. coli system, hepcidin is expressed as a reinsoluble form due to its complicated disulfide bonds. So because of these issues, to produce enough biohepcidin is necessary. So we start to think how to deal with these issues. We use another system to produce the hepcidin. We use the barfish egg as a bioreactor to produce the hepcidin. I introduced transgenic fish. So far, has already become the easy way to establish a transgenic fish. For using the total systems, more than 50% successful rate, you can get a transgenic fish. So in this case, we establish a recombinant plasmid. We call it. So first, I introduce what is ZP. ZP is Lona puricida protein. It's a female subphase glycoprotein related to reproduction in female vertebrate. Lona puricida only expressed in the oocyte. And in previous our study, actually we already used this promoter to express the fluorescence proteins in the barfish. So you can see GFP and IFP here is the oocyte. Okay, the arrows indicate the side is the oocyte. The fish just 1 month. So if the fish grow up to the adult, in abundance, all oocytes are GP or the IFPs. And here's how we construct a recombinant DNA. We use one Lona puricida promoter to trigger and the other Lona puricida promoter, we trigger the EHP. The GFP used as an indicator. So it means that if we see the fish with the GFP, it means antimicrobial hepcidin expressed. And the hepcidin in the C terminal, it tagged with the 6 histidines. It's for use the NINTA concurrent to purify. Easy to purify. So first, we establish the transgenic fish. So you can see here, this one is a wild type fish. It's a transgenic fish, but this transgenic fish only expresses the GFP. This transgenic fish, we use the GFP as an indicator. So when you see the GFP, you know that it expresses the hepcidin, but we still need to confirm that. Okay, in the upper leg, the F0, it means we inject the embryo and grow up. In the F0, we already can see the GP expressed. And after the mating and get the offspring, so it's F1. You can see have maternal express. So the embryo with the GFP expression. So it means the embryo, the egg also contain the peptide. So we collect the embryos and to check whether the egg, the embryo, contain hepatitic peptide. So we make sure that in 3 steps. First one, we use the genomic DNA as a plan and use the GFP or the hepatitis specific primer to PCR check. So you can see here that what only express the GFP and the GFP and the histidine. So you can see that you can get the band, but the thing only show in this changing phase. So this data means expression cassette already genomic integrate to the F1 changing lines. And second, we do RT-PCR, sure whether the hepatitis genes are expressed. So we do the RT-PCR. When we collect embryos, we analyze, we prepare cDNA and analyze use the specific primers. So you can see in the changing line, the hepatitis gene expressed. So it means actually the hepatitis genes is really expressed in the changing line. And finally, use the because we didn't have the hepatitis antibody, so we use to recognize, to check whether the hepatitis is expressed, and we also use the GFP, so you can see and confirm that the hepatitis peptide is expressed in the debuffing egg. Okay, so it means the egg contain the hepatitis peptides. We can use the egg as a material to do the experiments. The first, I check the antimicrobial spectrum. Okay, the antimicrobial spectrum, you can see here that A and the B is a Vibrio geneticus, and B is a masa, it's a antibiotic resistant pathogen. So you can see both pathogens are antibiotic resistant pathogens. So you can see here that if you use the ampicillin or kanamycin, it's not used. Antibiotic cannot kill both pathogens. But if we extract the crude hepatitis peptide from the fish egg and add in the agar plate, you can see clear zones here. Okay, this is a control. A is mean the control. It's also the supernatant extract from the fish. An egg just only express the GFP. It's a control. But if express the things, you can see a clear zone here. Okay, so it means this expression system, it can express a bioactive hepatitis peptide. And we also determine the against different pathogens. So here show the different aquatic pathogens for the Aeromonas, Streptococcus, or some one is Vibrio species. Okay, all pathogen species, antimicrobial peptide hepatitis can show the antibacterial activity here. And then we feed the debug fish the egg. We do the experiment. One is control. It means every day we feed the commercial dye. And what type control group is means without supplement the fish egg. And control means we supplement the liver fish egg, but it just only express the GFP. This group, ANHEPC, this group, it means in addition to the basal diet, we also supplement the barfish egg express the hepcidin. If the fish supplement with the egg, the hepcidin, you can see the immune parameters increase. So it means the hepcidin can modulate the immunity. It can improve the innate immunity. So we also do the challenge test. If the fish supplement this kind of transgenic fish egg, okay, it can increase the disease resistance against the pathogen infection. So here's also the good summary. Hepcidin was successfully expressed in transgenic fish egg using an oocyte superficie promoter, and the hepcidin expression egg activity against diverse gut-caustic pathogens. It means it has potential application of in agriculture. And the barfish supplement with the transgenic fish egg with expression the hepcidin not only increase expression of innate immune reaction, but also enhance the disease resistance to pathogen infections. Means this kind of egg have potential used in agriculture for biocontrol. Okay, in the third part, I introduce use the barfish as a model to study the antiviral mechanism of NMV, nervous necrosis virus infections. I think the previous lectures I already briefly introduced the nervous necrosis virus NMV. I introduce again. Okay, this symptoms after the groupers infected by the NMV. You can see the fish, the swimming behavior will unbalance or rotation. And if the status continues, it will cause the all poor the fish mass state. Okay, NMV is an important fish pest virus. Noda virus that target to the nervous tissues. Primarily is retina. It contains it's a non-envelope virus. Okay, and soft code shape, and it contains two positive strand RNA. One we call the RNA1, encoding the RNA. RNA2 encode the capture protein precursors. It means RNA2 encode the capture proteins. And groupers is a major host of NMV virus. Grouper is one important, most important economical fish in Taiwan. And NMV causes severe mortality of groupers at their larva stage and the juvenile stage. But if you check the schedule of NMV infection in groupers, in this stage, we call the NMV acute infections. It causes mass mortality. I remember that the mortality rate more than 90%, even 95%, more than 95%. But after the fish goes up to the juvenile stage or adult stage, status will reduce. It means the fish can survive. So it's no mortality. So in this stage, we call the NMV persistent infection. And why is so we use the buffish to study the mechanisms. So we back to the immune response system. It's a molecular signal. It's quite important for antivirus infections. For example, in the immune system, interferon activate. The MX genes can inhibit virus RNA, and interferons also PKR, the protein kinase, and the protein kinase can activate or phosphorylate the EIF2A, then inhibit virus translations. It's interferons, okay, it can activate the oligodenylate synthetase, and finally, it can cause the virus RNA degradation. So interferons act important roles in antivirus. And we check the buffish after the NMV infections. Okay, you can see we inject the NMV virus into the buffish as the different tissue and analyze the NMV virus. You can see in brain, okay, heart, liver, we can detect NMV infections compared to rat titers, percentage embryo survival and virus survival. If we inject the cause mass state, and if the survival fish grows up, we certify in the sections. You can see the many efficient symptoms in the tissue sections. You check the inner immune response after NMV injection. Okay, we find in the buffish larva that you can find in larva stage no MX and interferon expressed. Adult liver fish MX expressed and interferon expressed. So it means in the fish in the larva stage, the immune system still not mature. So when they undergo the NMV infection, the inner immunity is no functions. They cannot against the NMV infections. But after they become adult or juveniles, because their immune system really maturations. So after the NMV infections, they have interferon or MX this immune system to against the virus infection. So that's why they have the higher survival in this stage, in adult stage. So we also we can see the immune protein label adult larva. Okay, larva it's NMV infection. If you use antibody to analyze the EIF2 alpha, phosphor EIF2 alpha, no. But in adult, you can see the signals. So that's maybe the reasons why in the larva stage, once NMV infections and cause groupers high mortality in larva stage. So in here, we also do a challenge test, and in zebra fish, see here if only NMV infection is low, it's blue line here, okay, and if you just inject the PBS 2 days. It's a control. That's this pink colors. Okay, and if we inject the NMV and interferon at the same time, it's no function. It also just 2 days. After 2 days, the fish also days. But if we inject the interferon first, and after 2 days, then we inject we can see the fish, the survival rate is quite high and significant, different from the other group, the other 2 groups. So it means you can supplement interferons to the fish in the early stage. Okay, so then the fish have the can activate their immune systems. Then they can against the enemy infection. So in here's a mechanism providing the buffish is a larva stage and adult stage. When enemy infection, because at this larva stage, no interferon, so it cannot activate EF and alpha. So post the mass stage, it called acute infections. And adult stage, because the interferon, the immune system already maturation, already have interferons, they can activate the MS EF2 alpha. Cause the it means suppress the virus RNA and infect the virus replication, something like that. So it can survive. So it's called persistent infection. Okay, so we clarify the reasons why in the larva stage have higher mortalities, and why the adult stage have the higher survival rates, and we start to think how to prevent the enemy infections. So here's I show the 2 strategies. We call RNA vaccines. In here we establish a stable line in buffish, and the buffish can express the siRNA. Second, we developed a narrow lipoplex delivery system. Okay, we introduce the siRNA. I think the mechanism maybe all students already know that siRNA it becomes is a small RNA, and it can pair when you inject into the cells. The cell have the risk proteins. It can bind the siRNA and binding to the messenger RNA, your target messenger RNA, then cause the messenger RNA degradation. So it means we just say the NMV virus to RNA1 and RNA2. So it's an RNA virus. So if we can design a siRNA, and if the fish can express the siRNA, and the siRNA can target to the RNA, NMV RNA, cause the RNA degradation, then the NMV infections. So in our studies, in this case, we focus on RNA2. I just say the RNA2 encode the catcher protein. And in RNA sequence, we designed for siRNA. It have different numbers. The number means the sequence size. It's a siRNA sequence and the GC content. And after we transfection to the cell, to the cells. We make sure that whether the siRNA have function and cause the patients. So we find this one siRNA 730 have the high efficiency. So we use this one establish. We use this one to construct an expression vector, and the same the expression vector was inject to the debuffish embryo and establish the changing line. And then we analyze the changing line whether it can against the NV infection. It is stable line, debuffish. We use different virus titer to infect the changing debuffish. Per egg, the titer is 10 power 3, 4, or 5. We find if the titer is 10 power 3 in the F1 stable 9, after we infect the NMV, compared to the control. Almost the same, reach to the positive control group. But if we increase the NMV dosage, the survival rate reduced. Okay, and you can see the 10 power 5 becomes 0. So it means even the debuffish changing lines express the siRNA against the RNA2. If the fish undergoes a high dosage NV infections, protect potence still low. So, but if the in lower the titers, it can show the. Okay. But I think this titer is quite high. In a normal status, it can reflect. I think this titers can reflect to the normal status. And here we also use another ways to infection. Okay, this one we use the liposome formulations. I think I should introduce this one. If we establish the changing line belong to the GM, genetically modified fish, and you know that GM fish so far it have some problem because global consider it may be the cause the certain of wild fish, or if this kind of fish escape to the outside, it may cause the gene flow. So in practice use in aquaculture, I think the GM fish is difficult. And if you want to establish a GM fish in zebrafish, it's easy. But if you want to establish in a grouper, I think that it's quite difficult. So that's why we use another way. We try to deliver. We try to deliver another way. Use the orals. We prepare different formulations, and the formulation can deliver. Formulation can deliver the DNA, the siRNA, the vectors into the host. Okay, so they use the liposome and the lipoplex because some commercials. So I just say that okay, they contain liposomes. Okay, and they first time they contain the C-ray expression vectors. they feed. Okay, they feed the fish and check whether the fish have the HC rate is raised. They confirm after feeding, they confirm around 80%. They can find have the HC rate is raised. That means the formulation they can successfully deliver the DNA into the fish. So after use this strategy, they practice in groupers. Okay, so you can find it's control, vaccine control. Okay, and vaccine control fish number, the survival, I think the survival rate and challenge with the NMV. Okay, if the fish feed the vaccines, we call it DNA vaccine, it can increase the survival rate. Here's I do the conclusion. The acute infection in larva and jumeirah is largely due to the inactive interferon response. As upside to activate inner immune response during persistent infection in adult stage. The acute infection in larva and jumeirah is okay, so it's the same. Sorry, and finally is siRNA or DNA vaccine provides efficient way to reduce the NMV infection. For the research work, I share the research work here. And the final tab, the scientific writing tip for high impact factor applications. I just say that when okay, and can anyone listen to me?
Yes, sir.
Yes, sir.
Okay. I just say that when to allow me to talk about this topic, I quite uneasy to talk about this topic because the topic also my goals. I also wish someone can teach me how to publish a paper in good journals.
Oh no.
so today is I think the topic. I think I'm not a teacher the student to how to publish, but just shares my experience to submission experience to you. So the first thing when we publish papers, I think we should know you should find a journal, your target journal. It's quite important. Okay, and how to find your target journals? Because different journals they can divide into different categories. So first, the journals from the JCR Journal Citation Report Database. Like here's this journals. Okay, so in the Google, if you enter, you key the JCR or the Journal Citation Report, and you can key in the journal name. You can see the journal name and the impact factor here. The first, I think your target journal, you choose a target journal is quite important. So because of my research work, I know that when I do the survey and search, I find my research work it just belong to marine or fish water biology. These two categories. Okay. So first, I mean, okay. So first, I think you should choose a category based on your research field. And you can choose a journal that fit your specific work. Okay, so for me, my research work always belong to these two categories. And for example, if I want to submit the category, I find the journal agriculture is my target journal. Here I can see the impact factor 3.225. But even you know the impact factors, how do you know the ranking? How do you know the ranking? So you should entry to the categories. Then it will have the list. And the list all journal based on impact factor. So you can see here impact factor from high to low. So if my target journal is agriculture, you can see the number five. On the categories, totally have how many journal in this category. So I remember in this category, totally have the 56 or 59, 58 journal. So if the agriculturalist journal is belong to number 5, so this journal belong to the Q1 articles. So I think this is the first thing. You should choose your target journal. It's quite important. And then we back to research work. I think the 4 tips to publish a high impact factor journal have 4 tips. I just say the first. You choose the journal that the best your research work. So the fit is critical. Sorry, here I always have the noises. I don't know whether you can listen to me.
Clear, it's very clear, sir. I'm so sorry.
Okay.
Mute mic.
Okay. So fit is critical, because you should know one thing. The most famous journal might not the one best to your specific work. Choose a fit journal. It's quite important. Then back to the research work. Publish a good journal. You should do a good research and with solid data. Work, then you have chance to publish a good paper. But how to do a good research? I list some criteria. I think first it's essential to choose a good unit with a good mentor. Why I list these conditions? Because do research require equipment and need a good mentor to guide you. A good mentor will inspire us to find the best within us and guide us to a scientific success. The good mentor, it will teach you many things, and the good unit, they will provide many source let you to finish many. I think everyone knows that when you do experiments, you need spend money and you need equipment to finish your experiment. So if you want to finish a good research work, you still have the good unit with good mentors. Research when you do the research work, I think you should set your topic. But how to set a topic? So it means that you essential to read the lecturer. If you want to do research and you already have a topic, you need to read a paper. You should know so far what already be done new and what question is worth to address. Then read the papers. It's difficult to set a topic. So that's why I list the conditions. When you do a good research, you need to set a particular topic. But the topic, if you have ability to set a topic, you should read the papers and find the questions. After you read the papers, then you have ability to set an important research question, because you know, oh, okay, which problem or which questions is new. No one talk about this question, and know the research finish in this part. So it's worth to study. So I think read the paper and the set questions is quite important. And after you set the topic, you already know what question you need to address. That is design. Design is the most important aspect of any research study. Okay. Okay, design is quite important. Design is the most important aspect for any research study, because all editor look for the quality of the study design at the first parameters. So if your design is defect, so when you submit the papers, maybe the submission will not go through the review process. Maybe the editor directly reject the submissions. So design, the experiment design is quite important. And one thing, if your experiment design is have defect, it's difficult to progress further. Many projects are waste opportunity because of inappropriate experiment design. For example, the controls not used, something like that. It causes your data becomes not solid. So design is quite important. And after you design your experiment, I think you should confirm material, method, and experimental protocol correct experiment, then you can get the correct data. Although sometimes the data may not deserve data, it means sometimes the results not fit your predict. Only correct data can put hypothesis and can make a conclusion. I think the same in my lab, the students okay, they already have the topic and do the experiment. I always ask them before they do the experiment, I ask them to write down the protocol and detailed protocol, including the materials and the methods details. After we discuss one by one, and no problem, then you can do the experiment. Why I need to do this this kind of teachings? Because you should know one thing. If the protocol is wrong or something wrong, and you already spend time to do the experiment, actually the result, the outcome also wrong, also failed. So it just wastes your time and waste the budget, the money Okay, so for the master students, I think I think for the PhD students, I think it should have ability independent to think about the questions. Okay, but for the master students, if you can or you have many time to read the paper or the lectures, and the teacher maybe give you the topic, but at least I think in the experiment design, and check every step, every protocol is correct, then you do the experiment. Okay, it can help you to it's been no need waste time and let your experiments go smoothly. And after that, you finish all your experiments. Okay, and then you will start to write your manuscript. Okay, so you start to write your manuscript. It means you start to tell a story. The content must make sense. So first set a title. The manuscript title. The title is the first window for reader to look at your work. Therefore, select a title that can catch reviewers or readers' attention. Accurately describe the content of your manuscript and make people want to read further. So the title is quite important. When you write a manuscript, the title is quite important. The other one is abstract. Abstract also quite important. Okay, abstract represents a guide to the most important part of your manuscript writing content. Many readers and the editor, if you submit the paper, the editors will only read the abstract of your manuscript, then decide, go through the next step or reject the manuscript. So when you write the abstract carefully, it has to able to stand alone. And in abstract, you know that every paper the abstract is short, maybe just 250 text. But in this quite short text, what question should abstract answers? Your abstract have to address. I think it should contain this component. First, what was done? Your experiment, what was done, and what do you do it, and what do you find, and why are these findings useful and important? I think that this part you should conclude in your abstract. Okay, then it can attract the reviewer's attention. Okay, and about writing, I think in the writing, as to me, for me, you know that English is not a mother language. So writing when I'm writing the papers, I still spend many time to write a paper. But one thing you should carefully is when write a manuscript, demand clarity, brevity, and logic, especially the logic. I just say that when you start to write a manuscript, it seems that you start to tell a story. So each part should be logic, and use the simple language. So good scientific writing demand clarity, brevity, and logic, and each paragraph should be able to stand alone there and provide context to what procedure is and what follow it. And observe the rule of good grammar, spelling, and And the first thing that you must avoid any irrelevant information, no matter how strongly you lack it. What's the mean? You know that when you finish a research work, maybe you spend more than 1 year. You work very hard. So you want to write many things in your manuscript. But remember that no need about these things. Okay, so even you work very hard, but just focus on your research work. Your research work. Okay, so if any irrelevant information didn't add in the manuscript. And finally, when you write a manuscript, follow the instructions of journals. I think that every journal, when you check, they show the other instructions. And most common other instructions are including the title, abstract, introduction, material measure, result, discussion, okay, and reference. And then the figure tables, something like that, okay. But different journal, maybe the different type or different styles. So when you choose the target, already choose a target journal, then you should follow the instructions carefully to write the manuscript. And after you finish the manuscript, I think it is essential to review by a non-specialist college before you submit your manuscript. Important. Before you submit your manuscript, it's essential to appropriate. You have remember that you have only one opportunity to attract the attention. If this is waste by careless mistake or the omissions, your chance is lost. What do you mean? If one is receive your manuscript and if find many wrongs or many mistakes, even your research work, many content, and find a good answer for a new questions, the editor may not patient go through the next step and continue to read your manuscript. So we need to make sure you only one chance. One chance once you submit, you only one chance. So you should make sure your manuscript is well done. So let a non-specialist college to read the paper, to read your manuscript. Okay, specialist college because non-specialist college they manuscript and comment on the manuscript will like the readers. You know, if the same specialist, we sometimes we read the paper, we already know. So some part that we will careless or skip that point. Okay, but if the non-specialist college to read the papers, if he or she don't understand, then he will raise the questions, and then you can check the point, or whether it's a logic or not make sense or something like that. And the non-specialist can help you to check all mistake. Like the error, grammar, like that. So after you finish that, then you can submit. So sometimes we submit the paper. We always think that we can quickly submit the paper and quickly get the response. But didn't do well everything, every part, that's good. It also just waste time in the paper, maybe 1 or 2 weeks. If the manuscript didn't go through the next review procedure, still need 1 or 2 weeks. The editor reject the manuscript, and you still choose another one or another one journal need to improve your manuscript. So if that way, why you didn't work on the manuscript in the first time? Okay. So that's my point. Write your manuscript is quite important. It's quite important. You should prepare well in every part. Okay. And after you submit your paper manuscript, after you submit your manuscript, you will get a response maybe a couple months later. If your manuscript is rejected, it already goes through. No matter it goes through the review procedure or still in the editor's reviews. The same, I think the reason nothing more than the manuscript is inappropriate for the journal. It's lack the normative. It poses an interesting question. It has a poor or inappropriate starting design. I think nothing more than this reasons. But I think don't say that. If you get this kind of situation, don't say that. I often face this situation. So just improve your manuscript and try another journal again. The same. If the reason is poor, lack normative, or an interesting question, or inappropriate study design, then the question back to the originals. Just like previously I said, when you do a good research, you should read the paper. You should make a good question and good experiment design. Don't let the reviewer have the chance to challenge your fail, lack this kind of experiment design. Okay, so that's why in the beginning you should pay more time focus on your research work. Okay, and as the normative, I always think when you read the papers, all reflect to myself. Actually, when I do the research work, I know that it's difficult to find a new question. It's completely new. And sometimes, especially, we do the applications research work. Especially applications research work. Actually, many topic or many how to say that? Or many materials already be studied. But even you do applications study research, you should also find something new. Then you start to do this kind of research work. Not to do the complete the same research work. Okay, that's why you need to read the papers. Okay, then you know what already done before. sometimes you will see that in some applications papers, maybe just the fish species different, or just the material different, but the concept, it seems the same, but it's fine. I think it's fine. At least 1 or 2 points is new, and in your manuscript, you should emphasize the news. Okay, so for me, previous I introduced the probiotic. In my lab, I isolated some probiotic. Okay, when I choose a bacteria as a potential probiotic, I want to check whether the probiotic has been studied before. Sometimes the probiotic has been studied, but maybe studied in marine fish. Okay, so if I do in a fresh water fish, it's a small difference, but then you can emphasize this point in your manuscript. Or the probiotic news, no one study this kind of probiotic, then you can emphasize this point. Okay, so I mean when you do the research work, you should know this kind of research work, which point is new, and emphasize that. Okay, but even that, sometimes I submit the papers, the response the same. Yeah, it says lacking novelties. So I mean we try our best. And if everything we prepare well and submit to the journal, and the response is reject, don't say it's just submit to another journal. I think it's fine. It's fine. Okay, but if the response is a major revision, okay, the major revision, it means your manuscript have a chance for publication. Okay, so for me, so far, when I submit the paper and the response is major revisions, so far the result, the final result, all of my submission is accept. You need to follow the rules. What kind of rules? First, you try to provide the answer for all the comments one by one. Okay, and sometimes you will, when you read the reviewer's comment, you will find the reviewer did not understand your point, or you didn't agree their opinions. If you disagree with the reviewer's point, so you just state your opinions clearly. Okay, so sometimes reviewer will ask you to provide another experiment, but I think that's fine. Know that some experiment need more time to finish, but right now, if you tell the editor or reviewers that you need more time to finish, I think that they will agree. So I think if the review comment is like this kind of point, I think that is a quite easy point. It's easy to answer. And if you need supplement the data or experiment, you just continue to finish that part. If your response is a major revisions, I always think that you almost accept. Okay, until now, my submissions you know, the response can accept all the major revisions. But so far, my submission, the response are no minor revision. Every time, major revisions. But sometimes I check the reviewers' comment. It's quite easy to answer and not many points, but it's the same major revision. So it means when your response is a major revision piece, because I think the point, the reviewer comment, the point, you just follow the comment one by one to answer the questions. You always accept. Okay. So finally, I want to encourage the students. There will always be reward by giving. There will begin by making efforts. Try your best and do your efforts in research. You can get a good performance. Okay. So finally, thank you for your listening. I should say it's my lab, and today's the lectures work. Some parts, for example, the transgenic liver fish egg. Some parts finished by the reader, and some the rice hard silica. That's finished by the AC Lini and virus NMV. Some part I want to thank them to help me to finish some important research work. Okay, thank you.
Thank you, Professor Hu, for a very informative presentation. Moving on, now we come to question and answer session. Maybe Mr. Dailami can present screen about the question.
Pak Dailami, can you hear me?
Hello?
Yeah, Pak Dailami.
Okay.
Okay, sorry. Okay, thank you, Pak Dailami.
Professor Hu, the questions are already appeared on the screen. Maybe you can start answer the question. Thank you.
Okay. So let me check. The question was, how many percent is the SR of this liver fish when culture? Oh, you mean the survival rate?
Yes, sir.
The survival rate.
Oh, I can see. Okay. The liver fish we culture indoors because we have the fish runs culture indoors, and the survival rates are normal status. The survival rates are around 90%. 90% during your experiment period. Okay, for the control group, around 90%. How is the mass to identify good embryos? Okay, when the fertilized egg, we collect fertilized egg, and we will culture in a petri dish. After around 6 or 8 hours later, if the fish embryo belong to the bad embryos and the quality is not good, the embryo will become white colors. So we can remove the embryo. So when we do the experiments, the embryos, we will collect the quality of the embryo. But I know that sometimes when we do the transgenic fish, it needs inject at one cell stage. So and this time we just want to establish changing line. So we will inject many embryo, but just select the changing fish from the survival embryo. So at that time, we no need isolate the good quality embryo. We just inject all the embryo. But if we want to do the experiment in embryo stage, then we need to isolate good quality embryos. So we will, after the fertilization, we will collect the embryo. And after the 6 or 8 hours, remove the bad embryo. Let's start the experiment. Okay, and the second, there will be some question I want to ask, including why does this use simple embryo? Is there any difference when use adult fish? Oh, I think the audience maybe asked the rice hard silica. I treat the embryo and treat the why I choose 2 stage, because the embryo stage and the juvenile stage, the tolerance to the chemical, the tolerance ability is different. So sometimes you will find same concentration, the chemical concentration you treat in zebrafish embryo, the embryo is normal. It can hatching out and grows to larva and generally no problem. But if you use the same chemical to treat the juvenile fish, you will find at the same concentration, the same chemicals it causes the juvenile fish death. So it means different stage of fish they have tolerance ability to different chemicals or signaling. So that's why in our case we try 2 different stage fish be used for other fish, and what happened if like. I think it's good questions. Actually, right now I say the research topic we cooperate with companies, and the bio company already get a patent. The rice husk silica they get a patent, and they already use in other ornamental fish or some aquaculture food fish species in larva stage. So it can use in other fish, but one thing you should carefully dosage. I just say that if the dosage too high, it will cause damage. Actually, it will cause damage. So for each fish species, you should do the test. Then you can use. Otherwise, if the dosage is too high, it will cause fish death. Okay, if too low, maybe no functions. So I can make sure that it can use other fishes, no problem. But for each fish species, you need to determine optimize the condition. Again, okay, that's 2 questions. And 3 question is only affect immunity. Question 3. Show the question 3. Okay. It means what happened to inner immunity of generally the fish when RHS concentration more than 200. Actually, you will see the fish directly. And the fish, the swimming, when they cause the damage, you will find the swimming behavior is not abnormal. That's unbalanced, and they will just stay in the bottle of tank and didn't swim in and didn't eat. So it's very obvious. So if the dose is too high, based on their actions or behaviors, you already know it causes some problems. Okay, and secondly, it only affects immunity or it affects embryo stage or survival. Okay, I think the question is the same to previous. I mean, when you do this kind of compounds, you should measure optimal concentration, then you do the experiment. Okay. And optimal concentration maybe no function. I mean, maybe it cannot modulate immunity. It means when you check the immune parameter, maybe no response, but at least it's safe. The fish is safe. So safe is quite important. You make sure the liver fish or fish in the safe dosage. Then you can analyze the first function, the further functions. So I mean, here I think it acts whether it affects the immunity or it affects embryo development also. Actually, in safety dosage, the survival and development is no problem. It's no problem. Okay. And the question 4. Although change is quite popular, they are okay. Yeah, I just in the lecture I just said changing fish so far is a problem. In the research work, we can use the changing fish. It's no problem. So, for example, in my lecture, I use changing fish, okay, to study the mechanism against the NMV infection. I think it's no problem. But for the human consumption, so far, the only one case have been put by the FDA. It's a changing salmon. Okay. If the changing for the human consumption, you know that in the lectures in MPST, always students also ask this kind of questions. They ask me, okay, if one day, actually, it's already carried on. If one day you fed a changing salmon, ask me, teacher, you eat or not? I say because I learned and I know the fish. I always tell them based on the product. I mean, if the product has been heat becomes heavy treatment. For example, they have been boring heat treatment. The protein already denatured. So I willing to eat this kind of food. But if like sashimi, it's not cooked a product, I will not eat this kind of food. Okay. Oh, yeah. The second thing is how can we figure that the chilling fish is safe enough for human consumption? Actually, this so far this question have a certain answer. You know that if we want to further safety, it should be very long history, the eating histories, then you can evaluate. But now I think the global is the same. Every product, it can clear label, let you choose. Okay, I think the product may be cheaper than normal, but the transgenic GM fish may be cheaper, but it have a clear label, let humans to choose that you eat or not. But as the safety, I think that you should long-term history, then can have a conclusion. So far, still until now, I can give you the answers whether it's safe enough. Okay. So how is the mechanism I am implement it in Embryo? Okay, the mechanism is if in the zebrafish embryo, we use a transgenic fish. We let the fish express the siRNA. The siRNA they can against the NMV RNA. It causes the NMV RNA to the RNA degradation, so the RNA cannot action or function in the embryo. So the fish can survive. So that's the mechanism we use in transgenic fish. It's only affect embryo survival or affect immunity. Okay, interferon. If I think that if you in transgenic fish, if you express interferon genes, it will affect the immunity of the fish. I think that it's no problem. But if we express the siRNA, siRNA, the mechanism just to cause the NMV's RNA degradations, it affect the or it induce any immune response. Actually, I didn't essays. I can answer the question. But if we express the interferon in zebrafish embryos or in zebrafish, actually the interferon can modulate the immunity. Okay. The question 6, according the question is rice husk silica whether can conduct in a ship. Actually, in my experiments, I finished this part in zebrafish, and I just say the product it developed in, by Bell company. They also use in ornamental fish, ornamental ship, but not in aquaculture shrimp. So I think it can be conducted in a. It should be conducted in the shrimp. But the same situations, the same answer. I just say the same. How to say that? If you want to use in different species animals, you should make sure the rice husk silica is safe for this species. So if you want to use in the shrimp, you should make sure that which condition is optimal and safe condition and safe concentration. So it's quite important. So it's possible to use in shrimp, but you should optimize the concentration, then you can use. Okay, and further question? Okay. Because normally we put the title content. Question 7. He asked whether can give some suggestions about the content of the title. I think to write a manuscript, it also requires experience accumulation. So when we write a manuscript, let's back to the previous I said, do a good research work. Good research work. It means you should know your research work. What point is normative? What point is needed? Then you should emphasize that. Okay. So I think the titles are, it can sometimes for example, for in our lab, we do the applications research work. The normative sometimes is sometimes I should say something maybe without or have the normative because it just change the material or just a different material change. But if your research work face this kind of situations, you just complete to set a title that contain all part of your research work. I mean, all parts means what material you use. In what kind of species, and the conclusion. It means conclusion you should including the title. It means, for example, if your research work you find it can so you should say that okay, your materials can enhance material, enhance immunity or disease resistant, because your result, this is your result, and you want to let people know your material have this kind of function. So you should conclude, collect these terms in your titles. Give you this kind of suggestions because fields are very wide. Different research, they write the titles have different ways. So I just give you this kind of suggestions. Try to including your new point, your material, or your find in your conclusion. That's the point, especially your finding you should put in your title. Non-speech, do you mean that person had different wisdom? Yes, I mean the non-specialties college is mean your research work is different fields. Okay, it means that your research topic different. Okay, because when we write a manuscript, some point, because if you all belong your how to say that, both of you belong the same research field, and when you write a manuscript, some point, because of you understand that you miss, you write very easy or simple, okay, but you know who submit papers, the reviewer not maybe they feel not very close to your research work. So if when you finish your manuscript, if you can review by a non-specialties colleges, many questions. Some question may be easy, but it can help you to find at this point your manuscript can complete or input or help you to find some questions when the reviewer read your paper. So I think this part is important. Okay.
Thank you, Professor Hu, for the answer. Professor Hu, we have more questions from Faridatun Amalia Hasanah, but she wants to ask directly to you. Maybe Faridatun Amalia Hasanah, you can open your microphone and ask your question to Professor Hu.
Oke, Mbak Talita, thank you for giving me. Prof, maaf tuh saya mohon izin untuk menambahkan sedikit pertanyaan mengenai cara penulisan paper. Ya, thank you for the super presentation, Prof. Hu. In here, maybe I have one more question for you, but it's about the tips and trick how to write a scientific paper. So the question is, how to make our research be successful or maybe even implemented in society, because as usually something that is researchable is only done for testing, and maybe because it's not easy to be applied for the community. For the example, I had doing the research such as the intervention for the civic culture, and I think the results are significant enough to be implemented. But then it is not easy for the farmers to accept that, or maybe to implement it by them, or I don't know, maybe they are not interested, or because I don't have much money for them, I don't know. But I think maybe it caused by their habit. I mean, they are just familiar with the method they used before. So me or the other guys maybe here also have the same problem. Therefore, here we who are still sitting in student bench would like to ask you about tips and tricks about how to make our research can be successful or even implemented in society. Maybe you can sharing with me or with the others. Thank you.
Okay, because I'm not very clear, I make sure that your question is you already done the research work, but when you submit, or you know the many budget to submit to let the English pool readings, something like that, right?
Yeah. And
okay,
and how we can encourage our research that can be implemented for the farmers or the others community, something like this, sir.
The farmers?
Yes. Can be implemented.
You mean I don't understand what you mean. You mean the student?
Mm-hmm.
Encourage student what?
No, I mean how to make our research can be accepted by the farmers.
Okay. You mean how to make your research work accepted by the journal, right?
Yeah, and for the community, maybe for our lifestyle. I think for the research, the results of our research.
I would like to help sort of who.
Yeah.
I would like to help about the question from Faridatun. I think firstly emphasize to how can we submission the article to the journal first, I think. And the secondly, Faridatun have the opinion to the risk of the research to be implemented for the farmers.
Yeah.
Please, one by one, maybe. Please, your answer question.
Yeah, thank you, Prof.
Okay. Thank you, Faridatun. Please, Prof. Have you catch? Hello, Prof. Can you hear me?
Yeah.
Yeah. The question from Faridatun is firstly how can we manage our research to be submission in the high impact journal. Secondly is they have opinion that the result of the research can be implemented to the farmers. I think like that.
High impact journals.
Yes.
Actually, if you want to publish a high impact journals, I should say that I just previously said this also my goals. I also wish someone teach me how to publish high impact journals. But if you want to publish papers in high impact journals, have some first, well writing, not well writing, it means well prepared manuscript. It's required. And second, you know what called the impact factor. The impact factor, the numbers are 4 point, 3 point, or 1 point. What's the mean? It means the important. Because if it's important, many papers will cite your papers. So if you want to publish a high impact factor journal, it means you should review by yourself your research work. Any normative is quite important. If the normative, your research work have the 1 point, and with a normative, and the normative is important for everyone, or the question is quite important for life science or the agriculture, or can solve solutions one important issue. Then it can publish a high impact factor. So if you want to publish a high impact factor, that's why I just say you should review the paper, read the paper, and try to find a question, answer the question, and your data can solve these questions. Then you can publish a high impact factor. So that's why I just say in the applications field, in Q1 article, actually, I just show the ranking in marine and freshwater biology. The best article, the high impact factor, the impact factor is 7. But in the life science, sometimes I always more than 5, and high impact factor maybe 20 or then 15. So when we talk about high impact factors in our field, we call it agriculture or marine science. I think you just think you have some point. That point is no research study before. That's enough. That's enough. Or if you can find something like that, actually it's already high impact factor in our field or in this category. I give you one example. Last lecture in November 2, I introduced my research work, probiotic. Actually, probiotic already study in many you can see the many publications. They already talk about the probiotic. I can publish papers in I didn't say the high impact. I just say Q1 papers in this field because you should find something new. For example, if the potential probiotic study before I didn't do it. I try to select a new one, and a new. So it should be the new strand. Okay, and if the new strand have the new functions, then it can increase the so it can publish in the Q1 papers. So I mean you should review your research work. I think that when you do the research work, you already have ideas. In your research work, what is new? Your point. You should focus on your point. So when you write a manuscript, you should emphasize. Okay, then the journal can accept the point. Otherwise, for me, the probiotic function, the assay functions, it's just the same. For many published papers, it's also the same. But what is your new? So you should provide some new point or opinions in your manuscript or in your research work. I think that is quite important. Okay, so not just do the experiment. I don't like my students just know how to do the experiment and don't think about the experiment. Think is very important. Okay, so that's my suggestions. Back to your research book and review your whole stories. In which part is normative, it's new, and emphasize that. I think maybe it's a chance or opportunity to publish in the Q1 papers.
Thank you, Professor Hu, for the answer to the question. I think we should end the Q&A session because the lecture will be end at 11 a.m. So thank you, Professor Hu, for answers to all questions. Finally, we come to the end part of presentation. We would like to say thanks to Professor Xiaoyang Hu. Hopefully, the presentation will be beneficial for everybody. Thank you for your attention, and the agenda will be continued by MC. Thank you, Professor Hu.
Thank you. Thank you very much. Thank you, everyone.
Thank you so much, Professor.
Okay, thank you for Salita. Thank you very much, Professor Hu, for informative and insightful presentation.
Thank you. Thank you. Thank you very much.
We hope that this seminar can provide a lot of knowledge so that it can be applied properly and correctly. With the end of this seminar, the 3-in-1 Master Program of Aquaculture 2020 has ended. Thank you so much to Speaker Professor Hu, Head of Aquaculture Master Study Program, all lecturers, committee, and all seminar participants. Have a good day. See you at the next opportunity. Bye-bye.
Bye-bye.
Thank you so much, Prof. Hu.
Thank you, Prof. Hu.
Thank you, Professor Matthew.
Yes. That's experience.
Yeah.
Okay.
Okay, but
How about Marlinda, Prof. Hu?
Oh yes, yesterday she talked to me. Actually, right now it's normal in our lab.
Yes.
But a couple days ago, she talked to me that she still miss parent or the country. It means that she went back to Indonesia.
Yeah.
So I think I told her that no problem, just follow her heart. And you know that if right now its emotions, if it's on back, it's no. He can focus on his studies. So I agree. She back to Indonesia, and the flight is a Sunday morning.
Oh, yeah, Sunday morning. Okay.
Yeah, Sunday morning. And today she go to hospital to do the PCR check, the COVID-19, the PCR check. If can provide the check, she is no problem. Then when she back to Indonesia, she no need quarantine 2 or 3 days. I forget. So today she go to the hospital.
Yeah, I'm very sorry. I'm very sorry about Marlinda. I think we hope everything is okay for administrative and university, and also I have been to Prof. Lily also double degree in Universitas Brawijaya, and we also know about finally the state of the Marlinda will back to Indonesia.
So I think that is fine, no problem. But I think that right now that his status is no problems. It's healthy. No problem.
Yeah, because the condition of the healthy, I think. Yeah.
I think that we keep the contact. If any problem, any students problem, we keep contact each other's. Okay, and we will try our best to take care of the student from Peptide University.
Thank you so much. And I will send my email from you because of what they call it. There is some administrative about the bank. I will email you to complete the information about the bank.
Okay, no problem. It's email to me, and I will. Okay, I will answer to you. Okay.
Thank you so much, Prof.
Okay, thank you. Okay, bye-bye.
Nice.
Bye-bye.
Bye-bye.
Bye. Professor.
Bye-bye.
Bye, Professor.
Bye. Bye-bye. Bye-bye.
Oke, terima kasih adik-adik sekalian, luar biasa. Talita, good job.
Prof. Terima kasih, Prof.
Dayu, good job.
Terima kasih, Prof.
Terima kasih responnya, baik.
Terima kasih, Prof.
Ya, cuman memang biasanya begini, orang-orang luar itu biasanya kalau kita bertanya, kepengennya directly.
Iya.
Directly, ya. Tidak usah menyampaikan background-nya, tapi directly pertanyaan kita apa itu. Nah, nggak apa-apa, ini pengalaman, tapi udah bagus ya. Oke, sudah semuanya. Ini Mas Tri, Elizabeth, Pak Dailami, Bu Febri, Fadli, Zainuddin, Nabila Anggi, masih banyak lagi yang di bawah ini ya. Nur Alia, Nabila ya, Dayu, semuanya luar biasa. Terima kasih. Good job ya, pengalaman Talita sama Dayu ya.
Iya, Prof. Terima kasih, Prof.
Terima kasih, Prof.
Oke, bisa kita follow up itu nanti ada emailnya di Pak Maftub ya. Dia sangat bagus, sangat wise ya, sangat bijak, dan beliau sekarang jabatannya Dekan Biotek. Kalau di kita di MIPA ya, MIPA. Beliau adalah Department of Biotech ya, untuk di NPUSD, dan selama ini selalu minta mahasiswa ke Pak Maftub ini. Tahun ini sudah kita kirim 5, tapi ada 1 yang kemudian sakit dan izin pulang tadi, Mbak Marlinda ya. Dan tahun depan juga beliau juga sudah minta lagi, tapi definitively berapa mahasiswa masih belum tahu, nanti akan saya dalami dengan email-email dengan beliau. Oke, semuanya adik-adik sekalian, ilmu yang beliau sampaikan luar biasa. Hari ini kita jadi kesimpulan yang bisa kita dapat, namanya high impact journal itu tidak instan gitu kan. Beliau sampaikan tadi, ilmu tentang meneliti jurnalnya kita harus sudah kita teliti ya. Yang kedua adalah riset kita tidak boleh ya data di-shareable gitu kan. Jadi data-data kita harus fix data biasa gitu kan. Terus kemudian setelah itu punya kemampuan writing manuskrip ya. Dan yang terakhir, kalau kita sudah komunikasi ada revise. Pertanyaan saya terakhir tadi nggak apa-apa, nggak usah dijawab ya. Biasanya revisinya ada
tadi waktunya sudah nggak cukup.
Karena beliau pesan ke saya itu jam 10 mintanya. Nah, tapi nggak apa-apa, udah bagus. Jadi ada pengalaman saya itu minor satunya major, tapi majornya minta ampun jelimetnya. Jadi tapi pengalaman saya teruskan itu juga masih berat. Kemudian dia setelah revisi dia sarankan masukkan di jurnal ini saja, saya rekomendasikan. Akhirnya diterima. Oke, baik adik sekalian, dan penjelasan beliau tadi yang awal-awal itu memang mendukung pada bagaimana menulis jurnal yang bagus. Karena apa? Beliau menginginkan jurnal yang bagus ya berasal dari penelitian yang bagus, kan gitu ceritanya itu kan. Ada informasi RHS tadi kan, Rice Hash itu dulu diselesaikan oleh bimbingan saya, Desi ya, dari Banyuwangi, sama Rida dari bimbingannya Prof Yeni ya, dari Banyuwangi. Terus kemudian ada Antimicrobial Peptides, AMPS, kemudian hepsidin tadi, kemudian antiviral, kemudian kerja interferon tadi. Saya kira luar biasa ya, adik sekalian. Ini informasinya bisa kita perdalam. Nanti coba bagi yang berminat nanti bisa komunikasi dengan Pak Maftub, karena materi ini juga bisa kita sarikan dalam PDF gitu ya. Oke. Terima kasih, sukses selalu ya Pak Maftub, izin karena mau Jumatan ya. Baik, assalamualaikum warahmatullahi wabarakatuh.
Terima kasih Pak, waalaikumsalam.
Waalaikumsalam.
Terima kasih, Prof.
Terima kasih Prof.
Yuk.
Terima kasih, Prof.
Oke ya.